Say you take me. Now imagine I doubled myself. I could technically live a double life doing all the things I've never been able to do because of school or work. It's not as easy as we think. War on finding out how to clone began in 1952 with the nucleus of a frog advancing to a tadpole, with various of other testings in between, including cows, pigs, goats, rabbits, kittens, mules, horses, dogs, and possibly humans were made in 2001, 2002, and 2003. The first and most successful cloning was on a sheep named Dolly. They used the reproductive cloning. In reproductive cloning, researchers remove a mature somatic cell, such as a skin cell or an udder cell, from an animal that they wish to copy. They then transfer the DNA of the donor animal's somatic cell into an egg cell, or oocyte, that has its own DNA containing nucleus removed. Researchers can add the DNA from the somatic cell to the empty egg in two different ways. In the first method, they remove the DNA containing nucleus of a somatic cell and inject it into an empty egg. In the second approach, they use an electric current to fuse the entire somatic cell with the empty egg. In both processes, the egg is allowed to develop into an early stage embryo in the test tube, and then is implanted into the womb of an adult female animal. Ultimately, the adult female gives birth to an animal that has the same genetic makeup as the animal that donated the somatic cell. This young animal is referred to as a clone. Reproductive cloning may require the use of a surrogate mother to allow it develop of a clone embryo. Another type of cloning is therapeutic. Therapeutic cloning involves creating a cloned embryo for the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. These stem cells can be used in experiments aimed at understanding disease and developing new treatments for disease. To date, there is no evidence that human embryos have been produced for therapeutic cloning. The richest source of embryonic stem cells is tissue formed during the first 5 days after the egg has started to divide. At this stage of development, called the blastocyst, the embryo consists of a cluster of about 100 cells that become any cell type. Stem cells are harvested from cloned embryos at this stage of development, resulting in destruction of the embryo while it is still in the test tube. In November 2007, using a new cloning method that removes the egg's nucleus without dyes or ultraviolet lights, researchers produced the first primate embryonic stem cells. The work involved transferring the nucleus of a skin cell from a male monkey to the nucleus-free egg of a female monkey. These embryonic stem cells did not develop into a whole monkey, and researchers said their work was aimed at therapeutic applications. However, the researchers showed that with some adjustments, the technique used to make whole copies of other animals may also work in primates. Human cloning entails the creation of a different individual, a genetically identical copy of an already existing human being. Natural human cloning does exist and is commonplace, namely the existence of identical twins, which exist in today's society. In conclusion, cloning is very possible, but there is also an ethical and moral reasoning that they have not gone farther with it.